So this is the second of the independent study lab introductions. And in this lab we are looking at epithelium, which is the first tissue that you will need to be familiar with in your study of cell structure and function. The goals for the epithelium lab, in addition to what is listed as specific aims in your lab notebook, are to distinguish simple, pseudostratified, and stratified epithelia, and also to identify the elements of exocrine glands. This first image is a medium to high magnification light micrograph of epithelia, and what we want you to see here are three different types of simple epithelia. What we're looking at are cross-sections of the tubules in the renal medulla in a kidney, and I don't want you to be concerned about that particular detail at this point because we'll study the renal system later. But this slide provides a very good example of all three types of simple epithelium. Starting with the simple squamous epithelium, which I'm pointing to down here, please note that the nuclei are very flattened and elongated around this lumen. And compare that to a tubule which is just to the right of it. This is a simple cuboidal epithelium. The definition of simple cuboidal epithelium is that the height of the cell is approximately equal to its width. And you can see that the nuclei are pretty much central in these cells. And just up at the top, we have an example of simple columnar epithelium. Here we have cells that are taller than they are wide, which is the definition of columnar, and the nuclei are generally centrally to somewhat basally located within the cell. At the basal surface of these epithelia, you can see this dark pink line, and it is the basement membrane of the epithelium. The epithelial cells are polarized cells, as was discussed in the epithelial lectures, and having a base defines polarity for these cells. So here is the base of a simple columnar epithelial cell. At the opposite end is the apex, which faces the lumen, and then you have your lateral surfaces on either side. This is true for all epithelia. Even the squamous epithelia have a base and an apex and sides, though the sides obviously for the squamous epithelia are not going to be very wide. There are two types of structures that we can see here. Most of what you see are the tubules that contain the urinary filtrate, which cannot be identified. It's a simple liquid. Also here you can see blood vessels. So the difference here is that the lumen of the blood vessels are filled with blood cells, which you can see by looking at in the center of the lumen. So you can see four different epithelia. You have your squamous cells that are the endothelial cells, and this is specialized nomenclature for the blood vessels. Then you have your squamous epithelial cells for the thin loops of Henle, and then you have your cuboidal epithelial cells, and finally your columnar epithelial cells. The next slide that we're looking at comes from the epithelium of the small intestine. Again, please don't be concerned about the details of being able to identify where this is coming from. What we really want to focus on at this point is the ability to recognize epithelia, to name epithelia, and to begin to understand what their function is based upon their morphology. So initially, this epithelium looks more complex than the previous slide. In the previous slide, things were easily determined as being simple because they have a very simple outline that defines the cell. Here, life is a bit more complicated because we have other nuclei, which I will explain to you in a moment. So this is a simple columnar epithelium. It rests upon a basement membrane, which because of the staining of this section doesn't show up quite as nicely as the previous section, but nonetheless there's a basement membrane here. And then you can see the lateral surfaces of these simple columnar cells. And then at the top you'll note that it's fairly complex. There are multiple lines here that are showing up in this staining. Also, there are different types of cells. So there's the simple columnar cell, and then there's this other columnar cell which clearly has an accumulation of material in its apical cytoplasm. This is called a goblet cell, which secretes mucus onto the surface of the epithelium. The other cells are the absorptive cells in the gut. And at the top, at the apex, we have specializations that are called microvilli. These are structures that are too small to be seen with conventional light micrographs, but their presence can be detected by this thin blue line, which is actually the terminal web, which we talked about in the epithelial lectures. And this terminal web is the basis, is the insertion site for actin filaments, which then go up into the microvilli on the apical surface. And this increase in surface area is a functional adaptation to what the cell is here for, that is absorbing nutrients from the lumen. Now, the reason that this cell, or that this epithelium looks as if it is stratified, even though it's not, is due to these cells right here. These are T lymphocytes. There's one here, there's one here, there's one here, there's one there, here's another one. And you'll note that these cells do not touch the apex and they do not touch the base. So indeed they are not epithelial cells because they don't fit the definition, but they happen to be present within the epithelium, that is in between the epithelial cells, because these cells are looking for breaks which occur or could occur between these epithelial cells, permitting the contents of the lumen of the gut to pass this barrier and get into the blood in the connective tissue beneath. And that's something that is needs to be avoided. So these lymphocytes are constantly scanning through the epithelium of the gut to prevent any sort of invasion of bacteria or other material that's in the lumen that should not enter into the connective tissue space beneath. By comparison, the next slide shows a pseudostratified epithelium. How do we know that this is pseudostratified? Well, one of the first things that you should note is that there are two consistent layers of nuclei. There are the ones that are fairly oval in shape and are sort of in the middle of the cell or a little bit basal in the cytoplasm. And then there's another row of nuclei which are somewhat flattened along the bottom. These are the stem cells for this epithelium, and they touch the basement membrane, which is a pale staining layer just here, but they do not touch the apex, whereas the columnar cells touch both the basement membrane and the apex. So this is what we call a pseudostratified columnar epithelium, and we need to further define it, as you learned about in epithelium lecture, by the apical modifications that exist upon these epithelial cells. There are very long processes coming out of the apex, and these are called stereocilia. They are long microvilli filled with actin filaments, and they play a role of increasing the surface area of the epithelium, because what this epithelium is doing is resorbing some of the residual fluid and some of the cells and portions of cells that exist in the lumen. In the lumen, you will note there are somewhat unusual cells in appearance, and these are actually maturing sperm. This is part of the epididymis that you'll learn about in the spring as we study the male reproductive system. And these portions of these cells, or defective cells, as well as fluid and other things, can be resorbed by the pseudostratified columnar epithelium. You'll note at the junctions of the apical portions of the lateral surface of these cells, there are little points. And these little points are the junctional complexes that provide a very tight seal of one epithelial cell to the next to prevent anything from going in between the cells, which is called the paracellular pathway. And as you will learn about shortly, this epithelium sits on a connective tissue layer, which is underneath it. Note that there are a couple of blood vessels here, which come right up next to the epithelium. This is necessary because epithelium in and of itself is avascular, and it receives all of its oxygen and nutrients from the blood vessels below. And the initial layer of this connective tissue is what we will define as a loose connective tissue layer, which is cellular and contains small blood vessels, whereas just below that we have a more dense connective tissue layer. The next type of epithelium that we will look at now that we have finished with the simple and pseudostratified epithelia is what we call stratified epithelium. This image is from the esophagus, and it shows a multi-layered epithelium. So all epithelia are defined in part by the lumen that they touch, and here's the lumen of the esophagus up here. And what you will note in this low light micrograph, low magnification light micrograph, is that there are many, many nuclei. So these nuclei are very small spots. As we learned in the cell biology lab introduction, the magnification of the image you're looking at can be distinguished based upon the size of the nuclei, and because these nuclei are small spots, you know that this is a low magnification light micrograph. And what you will note is that there are many, many layers of cells, all having nuclei going all the way to the top. This is what defines a stratified epithelium. To name a stratified epithelium, we go to the uppermost layer, and that is part of the name. So this is a stratified squamous epithelium, and because it has nuclei, the cells are nucleated all the way to the top. This is also called mucosal. It's a wet epithelium, which is covered by serous and mucus secretions, which aid in swallowing food. Note that the connective tissue layer at the base of the epithelium is folded in to the epithelium. This helps to bring blood vessels and, of course, nutrients and oxygen closer and further up into the epithelium, which is dependent entirely on receiving its nutrients from the connective tissue space. There is a loose connective tissue upon which the basement membrane sits, and then below we have a more dense connective tissue with a large number of blood vessels. By comparison, here is a stratified squamous keratinizing or non-mucosal epithelium. This is the skin, and what you will note, starting from the basal layer where the stem cells are, as you progress upward, a significant number of layers of cells are nucleated, But then you get to this layer right here, which is kind of darkly stained. And at that point, the cells are making what is called the water barrier. And you'll learn about the components of the water barrier when we study the skin at the beginning of the spring semester. But beyond this point, all of the cells die. They actually lose their organelles and become filled with an intermediate filament protein that is called keratin. And the combination of materials made by these cells inside and outside, plus the keratin filaments, make this what is called a keratinizing epithelium. And the thickness of this keratinized layer is dependent upon the mechanical stresses that are experienced by this epithelium. So as you do some manual labor, for example, and develop calluses, you can think of this as increasing the number of keratinizing layers in this epithelium. As before, the epithelium basement membrane connects the epithelium to the underlying connective tissue, and you can find other things such as nerves. We won't go into this at this point, but there are nerve endings here that help in sensation of touch. So we've covered the the different types of epithelium: simple, pseudostratified, and stratified epithelium, reviewing what you have already learned in the epithelial lecture and notes. And now we're going to move on to exocrine glands. Exocrine glands perform a very important function in the body by secreting serous and mucus secretions. This particular image happens to be a medium magnification light micrograph of a salivary gland, and it has two types of what we call adenomiers or acini, which are the secretory units of the exocrine glands. So I'm outlining a mucus acinus or mucus adenomier, and then down here below are serous acini or serous adenomiers. Note that these are easily distinguished because the mucus adenomiers contain this light staining material, which is mucus ready for secretion into the mouth. And the nuclei of these adenomiers appear flattened along the basal surface of the epithelium. What's difficult to distinguish often is the lumen of this adenomier, and often you do not see the connection of the adenomier directly to the duct, which moves the secretions out onto the surface that this particular exocrine gland serves. By contrast to the mucus glands, the serous glands have more rounded nuclei in appearance. Again, it's difficult to see the lumen at the center of the acinus, but because the cells are secreting a serous or fluid secretion, they have a darkly staining appearance because of the enzymes and other substances that are found within this serous secretion. Another thing you should note, at the base of these adenomiers, you will find these flattened looking cells. These actually exist inside the basement membrane of the adenomier. These are called myoepithelial cells. And in the next slide, these myoepithelial cells, because this is a higher magnification light micrograph, stand out more easily and are more easily recognized. So the arrows are pointing to these flattened nuclei that do not touch the lumen of the epithelium. This structure that I'm outlining with the arrow is a duct, and you can see the lumen of the duct, and it goes out of the plane of the section temporarily and then comes back in, but this is likely all one continuous duct lumen. And the duct is responsible for transporting the fluid from the adenomier to the surface that will receive the fluid. And the myoepithelial cells are contractile cells that exist within the epithelium, even though they are not epithelial by nature. And when these cells contract, they aid not only in squeezing the fluid out of the adenomers, but also helping the fluid move through the duct towards the opening of the duct onto the surface that this particular gland serves. So that's an introduction to the epithelium lab. What I would like you to do now is to go to the lab and look at all of the images. Now that you have learned how to recognize the epithelium, you can test yourself as you're looking at these images and reading the brief text that goes with each image. Once you have looked through all of the images which complement what you have learned in the lecture and from the course book, then you can test yourself in a couple of ways to see how well you understand the material. One of the ways to do this is to go to Learn at UW and look at the image quizzes, a quiz or quizzes that are available there, so that you can identify cells, epithelia in this case, and other structures such as goblet cells and lumens and things like that in short quizzes that exist on Learn at UW. Later in the block, as you are preparing for the exams or the quizzes, what you should do is use the unlabeled images in the histology atlas to test your knowledge, and then you can check your answers by going and looking at the labeled images, which are also part of the histology atlas. And finally, if you can work together with a small group of friends, say 2 or 3 other students, you can either look at images on a laptop or project images and test each other with these unlabeled images, asking each other questions about cells and cell organelles and tissue structure that you see in these images.